Hello, am I audible? Hello, Amol sir.
Ah, sir, should we start?
Yeah, I'm ready.
Yeah. Is my screen visible? Can someone acknowledge?
Yes,
Yes,
sir.
we can see.
Okay, so good morning, all. Good morning, Amit sir. I'm very privileged to welcome you for this FDP on Biomedical Engineering Systems and Technologies. So I would like to introduce Amit sir to all the participants before going ahead. So Dr. Amit Sethi is our speaker for the first session. He is currently a faculty member in Electrical Engineering Department at IIT Bombay, and he is also adjunct faculty at Department of Pathology US. He has done MS and PhD from University of Illinois at Urbana campaign. He has served in capacity of assistant professor with IIT Guwahati and as a consultant with ZS Associates Chicago. His research interests are computational pathology, medical image analysis, machine learning mainly, and several others. His recent research is topics are like metrics in breast cancer, open source algorithm to color normalize histopathology slides, image super resolution, detecting subclonal mutation, and so on. Uh, his research groups are working in various of these research areas. Among five finalists for NVIDIA Global Impact Award in 2017. So this is also one of his credentials. So with this brief introduction, I would like to invite Amit sir to start his session.
Thank you, Mr. Amol. I'll share my screen now.
Yes, sir. I will stop presenting. I request all the participants to turn off their camera so that we can save the bandwidth.
Good morning, all the participants. I hope you can see my screen.
Yes, sir. Your screen is visible.
Okay.
Good morning, sir.
Good morning. Thank you to Mr. Deshpande to invite me and for this nice introduction. So I work in IIT Bombay in electrical engineering, as he mentioned, and my work has a lot of overlap with computer science and also pathology and radiology. I use artificial intelligence to analyze images primarily of cancer and primarily of pathology in cancer, but I also do a little bit of work in radiology and clinical images. So the way things work in pathology currently is that you see this picture on the right. People are looking inside a microscope, and a glass slide is there on the stage, and there's light that is shown below the microscope. And people look at the pathologist will look at the pattern of the tissue, how the cells look, how the cells are arranged, and based on that, if they see abnormalities, those could be signs of cancer. And if there is like more abnormality, then it is high grade cancer. If it is less abnormality, then it may be like pre-cancer, what is called hyperplasia and so forth. So the idea is that here, first of all, it takes a lot of time for pathologists to look at these slides. And this time taken can cause delays in resource constrained settings, such as in India, where we don't have a whole lot of qualified pathologists in remote locations and so forth. So one solution to that is telepathology, where you just you fix a camera to these microscopes, and the microscopes are manned by technicians, not pathologists, but the image is sent via the internet to a remote located pathologist who might be in a bigger hospital, and they can do the diagnosis. But it still doesn't completely solve the problem, because The number of pathologists themselves are small. And if you overload a pathologist, they will start making mistakes just like any other human who is overworked. So, and then there are certain very subtle changes in tissue that humans are not very good at quantifying. So instead of that, the solution that will start to roll into clinics in the next 5-10 years would be use of these digital scanners. These are currently expensive machines, but the new inexpensive ones that only cost, let's say, 10 lakh or 20 lakh are starting to come in. Before that, they used to cost 1 crore or 2 crore. And what they do is basically they scan the tissue with very high magnification so that you can see the nuclei and the cells very clearly on the screen. And once you can see them on the screen, that means it's a digital image. And that digital image can be analyzed using machine learning. And this machine learning works primarily using the same technology that you have in Facebook or Google Lens that can recognize your faces, that can recognize objects. But instead, that same technology or similar technology has been repurposed to recognize signs of cancers and various subtypes of cancers, because the cancer treatment now is moving towards, or it has already moved towards, precision diagnostics and precision medicine. What that means is you don't treat all breast cancers the same way. We look at different molecular markers in the cancer, and based on that, the chemotherapy drugs or targeted therapies or immunotherapy, those are quite different for different subtypes of cancer, even if they are from the same organ. So here, how AI is helping, I'll give you some examples from my own research. Okay. So before that, a quick introduction of machine learning. So a lot of machine learning, what we call machine learning, is actually supervised machine learning. What that means is that we have some training data. So we take, let's say, 1,000 cases where we know that the outcome was good after the treatment. So let's say we take 1,000 cases of cancer, and we know, 500 of them where the outcome was good. What that means is that the patient survived without disease after the treatment for 5 years. And let's say there are another 500 cases where the outcome was bad, which means within the 5 years, either the cancer came back or the patient died. Okay, so let's say we have all these red dots which represent the patient that had worse outcome and blue dots that represent patients that had good outcome. And we look at two features. So initially at the time of diagnosis, when we were looking at their cancer cells, what was the radius of the nucleus of the cancer cells, and what was the size of the gland that the cancer cells were forming. So if the glands were large, that means it is a good outcome. But if the nuclei are large, then it could be bad outcome, because enlarging nuclei are signs of like some sort of abnormality. So based on that, you have these red dots and blue dots that represent good outcome and bad outcome. And the job of machine learning is to find this black boundary that separates the red dots and blue dots. And you will see that not every red dot and blue dot is perfectly separated, because just these two factors may not be enough to make a perfect prediction. So no machine learning is perfect, and no machine learning is also, so we should be careful about taking neither of the two extreme views, which is that machine learning can do everything. No, it cannot do everything. It will always have errors. And the second extreme view is that machine learning will never be able to do a human's job. Humans take several factors into account, and the human expertise of a pathologist and radiologist cannot be replaced. Even that view is not completely true. For very well-defined tasks, machine learning is now becoming almost as good as humans, and sometimes even better. But I will caution to say that it is only for very narrowly defined tasks. So, for example, if a machine is trained to look at only invasive ductal carcinoma in the breast cancer, and you suddenly throw ductal carcinoma in situ, a different type of cancer, to it, then it will not be able to judge the difference between that. It is seeing something new. So that way the machine is not very intelligent, but for pattern recognition, it becomes very, very good for a very narrowly defined problem. So this is just a toy example where we are looking at only two factors, radius of nucleus and size of the gland. But in reality, you would look at thousands of factors. So your this equation of this black boundary that you see, that equation will be very, very complex. And that equation is captured in a neural network, which is a very complex equation. And it has millions of different parameters that go into defining that equation. Any questions? All right. Let me check the chat. Okay. So now let's look at certain characteristics of cancer that we are trying to classify using machine learning. So cancer is a heterogeneous disease, like I mentioned earlier. Not all cancers are the same. So even if you look at a single organ such as breast or lung, right? So not all patients have the same type of breast cancer or the same kind of lung cancer. They have different mutations that can cause cancer. There are many mutations that happen every day in our body, and most of these mutations only lead to an abnormal cell that dies on its own, and it doesn't lead to cancer. Once in a while, we have mutations that give the cell a advantage over other cells, and it starts to multiply and it doesn't die. It is not killed by the natural killing uh, mechanisms of the body. So that becomes cancer, and that chance of becoming cancer is higher if people smoke, if they drink alcohol, if they don't exercise, if they take too many artificial chemicals as pesticides and so forth in their food and in air pollution and so forth. So all these contribute to these mutations, but the mutations can be very different for different cancers, even within the same type of cancer. What is even more challenging is that sometimes there are two mutations in the same patient, in the same organ. So the cancer itself within the same person is heterogeneous, not homogeneous. So we are talking about intra-tumor heterogeneity, not just inter-tumor heterogeneity. It's not just between different patients. Within the same patient, one portion of the tumor can have one mutation, another portion of the tumor may have a different mutation. So the problem with this intra-tumor heterogeneity is that if we treat the cancer assuming one major mutation, the the secondary mutation may leave the cancer untreated and it will come back with a bigger force after the treatment, maybe a year, maybe 2 years after the treatment. So that's why it is now important to detect all kinds of mutations so that we can give people better treatments. So these are some examples of studies that others have done, how intratumor heterogeneity has been implicated in treatment failure and in poor survival. So we first started looking at intratumor heterogeneity not using images but by using gene profiles. The challenge that we faced was that no one had defined in mathematical terms what is heterogeneity in cancer. So if you look at this cancer slide, right, so each black dot that you see is a nucleus of a cell, and it looks pink and purple because some dyes have been put on it, and these dyes make the nuclei look dark purple, like the black dots that you see, and the pink portion is the connective tissue, the stromal tissue. So here, what you will see is that, so let's say if we do gene profiling of whatever is the tissue in this yellow circle, you will get certain different genes will have different gene expressions. So this is not exactly the same for all patients. It's slightly different for every patient, primarily because of their own inherent genetic differences and also because of how the mutations in the cancer have progressed in each patient. What we have, researchers have done previously is that within breast cancer, they have defined four major subtypes: luminal A, luminal B, HER2, and basal breast cancer. And they match the patient's mutation to whichever is the closest match. It may not be an exact match, but the closest match. And based on that, they will give the treatment that is most effective for luminal A breast cancer, as opposed to the one that is effective for HER2 breast cancer. But what can happen in a patient is you can have two different regions of cancer that have two different mutations, and sometimes those get left untreated. So, and that can be seen in slides when we put special chemicals such as this HER2 new stain to find HER2 protein. We see that in on the right hand side, you can see only some of the cells are brown, the others are not brown, because only some of the cells are overproducing this HER2 protein and they are detected, others are not detected. So that means even within the same cancer, you may have some HER2 positive breast cancers and some regions that are HER2 negative. So question is, can we detect multiple mutations in the same cancer? So that's what we started to do. We started to look at each patient on the HER2, low HER2 and higher HER2 axis, and low basal and high basal characteristics. So do their images look more like HER2 or more like basal breast cancer? So this is using a data from the Cancer Genome Atlas, which is an open source data set produced by the US government. And so they have released a lot of genomic data as well as image data. So we started looking at their genomic data. So the microRNA data, the reverse protein phase array data, and the DNA methylation data. And what you see, the green and the red dots are basically each patient is a dot in the three graphs. And we started looking at their top two distinguishing features for miRNA, for RPPA, and DNA methylation for HER2 versus basal distinction. So whether the breast cancer is HER2 cancer or a basal cancer. And what you can see is that all the red ones are tightly clustered, all the green ones are separately clustered, which is encouraging. But we also see some green patients that are actually intruding into the red cluster and vice versa. Some red patients are intruding into the green cluster. So the question is, what is happening with them? So our hypothesis was that these are the ones that don't have a single mutation, but they have multiple mutations. These are ambiguous or heterogeneous cancers. So we looked at all the ones that were away from the boundary, that were only within their own cluster, and we trained machine learning models to recognize their visual patterns. So this is a zoom in of a slide. And so each of these black dots that you see is a cell nucleus. And these are the blue patches that I show is what is the input to a machine learning model. The machine learning model looks at different sub images of this very large slide, and it models the pattern based on the label that we gave to it, which is whether it is HER2 or basal, or whether it is luminal. So this is the modeling process. Basically, from each patch, we detect all the points of interest, which are usually nuclei. We mine their texture features around that, and then we train a classifier to classify between high HER2 and low HER2, and then high basal and low basal. So once we did that, we tested it on held out patients, and we got some very good results. So the green dots again are basal, and what we found was that patients that were purely basal, they were high on the basalness axis. Patients that were purely HER2 were high on the HER2 axis, and the luminal patients were low on both axes, which is what we expected. Whereas the patients that were intruding into the other cluster, they came out in the middle. That means they had a mixture of HER2 and basal signatures. So this was quite interesting. It was a proof of concept that heterogeneity can be detected uh, using machine learning on pathology images. Uh, So based on that, what we can do is now we can take different sub-images and give it a score of whether it is HER2 or basal and produce a heat map. So with this heat map, you can see that some regions are more HER2-like than other regions. So the red ones are more HER2-like, the blue ones are less HER2-like. And we can do the same for basal. And if you see like non-overlapping red spots for HER2 and basal, that means it's a heterogeneous cancer, and it should not be treated just for HER2. It should also be treated for how you would treat a basal cancer so that it doesn't come back. And we got a visual proof when we started looking more closely at the patterns of uh, of how they form the gland. So if we just look at uh, homogeneous cancers, HER2 looks like what you see on the top right, and you can see that the HER2 patches from heterogeneous tumor look somewhat similar to them, which is on the top left. Similarly, for basal, the cancers look like what is on the bottom right, and the patches that look like basal from heterogeneous tumors, which is on the bottom left, they look similar to that. So the problem of defining heterogeneity as a metric in cancers, that still remained unsolved by the time we had done that work. So we thought, okay, let's go back to the genomic data and try to mathematically define heterogeneity instead of that ad hoc approach where we look at visually whether the one particular patient is intruding into the cluster of other patients. So we started looking at again, it's called PAM50. It's a well-defined set of 50 genes to characterize breast cancer. And we plotted some 1,000 patients on these 50 dimension. It's just shown as a 2 dimension, but it's actually a 50-dimensional plot. So that dimension reduction is done using some tool called tsne. And what we see is that all the blue dots that you see, these are all luminal B patients. All the green dots are HER2 patients. The black ones are basal patients, and the red ones are uh, sorry, the black ones are luminal A and the red ones are basal patients. Um, so what we see here is again the same kind of pattern, that the clusters are kind of different from each other. They are different. They are spread out from each other. But then there are patients that intrude into the other clusters. So we defined a metric for this based on something called Mahalanobis distance. We looked at all the cluster centers, which are the big dots, and based on that, we saw whether a particular patient, what is the ratio of its distance from its own center versus nearest other center. And based on that, we defined three sets of patients, which are pure, which means they have only one type of cancer, or they have admix, which means they have more than one type of cancer, and neither means that they are somewhere between the two. There's some evidence of different two different mutations in them. So, and then we looked at their uh, survival for luminal A breast cancers. So the pure luminal A are the blue line, and the admixed luminal A is the red line. So what you see here is something called Kaplan-Meier curve, where your time is shown on the x-axis and survival probability on the y-axis. So initially all patients are surviving because that's why we are treating them. So you will see survival probability as 1. But as time progresses, as patient starts to die off, you will see that the survival probability of that group is going down. And ultimately, after several years, of course, everyone is dead, so it will be 0. So you can see that if the curve is above than below, that means that group of patients has a better probability of survival. If the curve is below, then that other group has a poorer outcome on an average. So you can see that the pure luminal patients, they have a much better chance of surviving than those that are admixed with some other kind of breast cancer. They have luminal as well as something else, such as basal or HER2. So this was good proof of concept for us that our metric for defining heterogeneity is working. And we looked at the metric in another way and we got similar results. What is more interesting is that there were actually differences in the pure versus admixed patients at the molecular level and at the clinical level. So if you look at clinical differences, the admixed patients, their age on an average was higher, 64 years versus 59 years. And which makes sense because as the person grows old, she accumulates more mutation. So the mutations will be of different types. So the admixed patients have usually older age compared to the patients that have only a single type of breast cancer. Okay. Similarly, menopausal status, premenopausal status is higher for the pure breast cancer patients, pure luminal patients, and which is also correlated with age. The younger women are premenopausal, older women are postmenopausal. And then proliferation is also lower for 65% of the women among the pure have lower proliferation. Proliferation is a set of genes where which indicate how fast the cancer is spreading. So they have lower proliferation, uh, and their tumor sizes, that is not important because these two are quite similar numbers. There was no difference in tumor size. So which is also important, even though the difference is small. Uh, in fact, it is important that the difference is not large, which means that the effect that we are seeing in survival is not because of the size of the cancer. Okay, the effect that we are seeing is because of other things such as the heterogeneity metric that we have defined. And then node positivity is whether the tumor has spread to lymph nodes or not, and that is smaller for the pure breast cancers versus the heterogeneous luminal breast cancers. Similarly, when we look at molecular differences, there are differences there. So, for example, high proliferation by ORCA is higher for admixed women, right? And their recurrence score by this PAM50 gene panel is also higher for the admixed patients, which is what we expect, and that is what is causing their poorer survival. Okay. So this tells us that even though we get, let's say, 500 women, all of them have luminal A breast cancers, we will be able to, by looking at their genes, we will be able to tell which ones of them have pure luminal breast cancers and which one of them have admixed luminal breast cancer. The ones that have admixed luminal breast cancer are less likely to respond to treatment and survive long term. So probably the treatment given to them should be more aggressive to save their life. And the treatment should not be unnecessarily aggressive for those that have that have a treatable breast cancer, easily treatable breast cancer, because treatment has cost associated, discomfort associated with it, and quality of life degradation associated with it. So now what we want to do is that now that we have defined what is heterogeneity in breast cancer, we want to now use uh, deep learning methods, deep neural networks, to detect different foci of luminal A, luminal B, basal, and HER2 breast cancers in an even more accurate manner than what we did in the previous study. So here we started looking at HER2 breast cancer first. So what you see on the left is a special type of a dye that is put on the tissue that detects HER2 protein, and it becomes brown when it detects HER2 protein. But this special dye, it's called HER2 IHC, and this one is not easily accessible in most pathology labs, and it is a little more expensive test, whereas the one that you see on the right is an inexpensive test which is available in every pathology lab, which is called HND stained slide. It is not a specific slide because specific stain, because it does not tell you whether the cell is HER2 positive or not. It just shows where the cells are. So question is, can we use deep learning methods which are easily replicable on a computer and use it on the images that are on the right side, which are inexpensive, and based on that detect the results that you would need an expensive test to do. So this, if you just look at the slide on the right, most pathologists will not be able to tell whether it is HER2 positive or not. Some of them will get a intuition that it looks HER2 positive, but they will not be very sure of themselves. Whereas we show that with a machine, with deep learning, you can actually detect it very accurately. So this is where I was talking about, that if the problem is narrowly defined, deep learning or machine learning can actually do a good job of detecting these subtle changes in the tissue. So how we did that was that we looked at these slides that you see on the left, the HND stain slides, and we marked regions that have cancer, which is this blue region, and the regions that don't have cancer, which is the green region, and we got it checked by a pathologist. And on the right, you see the corresponding another section of the tissue, which is stained by a HER2 stain, and you see that the staining is light brown. That means it is not HER2 positive. It is HER2 negative. So it's not a HER2 positive breast cancer. The treatment for HER2 positive breast cancer is very well defined using a drug called Herceptin, which has been in the market for 10-15 years. Before that, HER2 uh, was a breast cancer with a very poor survival, but now the survival has improved because of this drug that has come into the market for the last 10-15 years. Even in India, it is the standard line of treatment for HER2 positive breast cancer. So the advantage of detecting this on the kind of slides that you see on the left is that even if the protein is not coming out of the cells to stain it brown, or if the brown stain is not available, the IHC is not available, using a computer, we'll be able to detect whether there are signs of HER2, and because of that, whether to give the person Herceptin or not to save their life. So, and then the slide on the bottom that you see is intensely HER2 positive, and that has been marked by this pink boundary that you see. And the corresponding section you see on the right is the HER2 stain slide, and then you can see that it is intensely brown because it is HER2 positive. So what is this deep neural network that we use on these slides, right? So this is the same technology that has been that a lot of big companies are using for other kind of image analysis. So for example, Facebook detects your faces and the images that you upload, and Google Lens detects objects that you point out, and then it searches in Google Images for similar objects and so forth. So those kind of things can also be used for pattern recognition on biomedical images. So essentially, what you have in a single neuron in a very simple neural network is that you have different inputs x1, x2, x3, and you multiply them with some weights w1, w2, w3, and you add them up. So you have x1 w1 plus x2 w2 plus x3 w3, and then you put it inside a function and you get an output, right? So that output looks like uh, a plane in this 3D space of x1, x2, x3. Uh, and that separates, let's say, one type of cancer from another type of cancer, where x1, x2, x3 are three different inputs about the cancer. Okay. So, but this doesn't help us make very complicated boundaries between two different types of cancer. So what is done is, first of all, to train it, we look at the desired output versus the output of the neural network, and whatever is the difference between them, we use that to tune the weights. But to make the thing more complicated, usually we add another layer in between and several layers in between. So with these hidden layers, the boundary between two different outputs or two different cancers becomes more complicated. And that's what we want, because a simple linear boundary like a plane or a line is not sufficient to do highly accurate classification between images of different cancers or between genomic signatures of different cancer. So usually there are too many several layers that can be added between the input and the output layer. But then it becomes very, each of these arrows that you see has one parameter associated with it, which is unknown, and that has to be, we have to optimize the neural network to find that parameter, what is a good value for that parameter. And it becomes too many parameters, and it becomes a humongous problem to solve it if your images can be very large in number. So if you're looking at image pixels being input like x1, x2, x3, so for a 1,000 by 1,000 sized image, you would be inputting 1,000,000 different pixel values. Actually 3 million, because you have red, green, and blue channels. So that's a lot of input, and then the number of weights become quite large. So back in 1998, Jan LeCun came up with a trick, and before that, someone else also came up with a similar idea, that why don't we repeat the neuron in the hidden layer? So if we take three different inputs, so let's say these are three different pixel values, they go into a neuron, and that same neuron is replicated for three other inputs and for three other inputs and so forth. So you can see that the same neuron is being replicated, only the inputs are changing. So this operation is called a convolution operation. And this convolution operation is similar to the convolution operation that some of you might be familiar with in signal processing. So because of that, you don't need new parameters. The same parameters, the black, the green, and the red arrow that you see, can be used again and again. So that reduces the size of the number of parameters of the neural network, and it helps us put more layers between the input and the output. So now the current state of the art is that some of these neural networks have hundreds of layers, some have 50 layers, some have 30 layers, and so forth. So a lot of layers. Before that, the norm was, before 1998, the norm was that you can only have one layer, at most 2 or 3 layers between the input and the output. And now we have around hundreds of layers. So the other change that has come in the field since 2010 or so is 2012 rather, is that people have started using GPUs, graphics processing units, and these are cards inside a computer which have thousands of cores. Like your computer has 4 cores, 8 cores, 6 cores, 12 cores, right? But the computer CPU cores are more capable. These are thousands of cores. They are less capable, but because there are thousands of them, they can do simple jobs in massive parallelism. So because computing the output of the same neuron at multiple locations is a parallelizable operation, you can send it to different cores. So GPUs help speed up this operation quite a bit, specifically NVIDIA GPUs that are currently being used for deep learning. And now there are special libraries on top of that, such as TensorFlow, CUDA, TensorFlow, Keras, PyTorch, that help you program to utilize these GPUs without having to worry about how to send different computations to different cores of the GPU. That is handled by the library. You just write a code in a very simple way, and it takes care of the rest. It also takes care of the differentiation of the output with respect to the weights, so that you can learn the weights to maximize the output, to maximize the accuracy at the output. So we use these neural networks, and in this pipeline, coming back to this HER2 positive breast cancer detection, we use 3 different neural networks to accomplish the job. So we take the entire whole slide, and by the way, this is a very large image. The size of this image would be 50,000 by 100,000 pixels, which is gigapixel, not just megapixel. So from here, we extract different sub-images, and from that, the first neural network detects all the. So if you see all these white dots, these are the nuclei, which correspond to the black dots in the image on the top left. Can you see my cursor?
Yes, sir.
Okay.
Yes, sir.
We can see your.
Okay. So our first neural network just does one job, which is detect all the nuclei. It is specifically trained to do a very narrow function, which is just detect the nuclei. Don't make any decision whether they are cancerous or HER2 positive. Just detect the nucleus. Then the second neural network filters those nuclei into only the cancer nuclei. So it detects only the cancer nuclei, the cancer cells. Then the third neural network takes the cancer cells and detect them into HER2 positive or HER2 negative cancers. And the output that you see is something like this. So on the bottom panel, you see again those inexpensive HND stained images. On the top panel, you see the expensive HER2 IHC images for the same tissue. And the blue dots are cancers, but they are not HER2 positive. The green dots are non-cancer cells, and the red dots are cancer cells that are HER2 positive. So 3 different types of cells it has detected. First of all, you can see that everywhere you see a black dot, which is a cell, a nucleus of a cell, there is a detection dot, which is either red, green, or blue. So that means our cell detection algorithm or a nucleus detection algorithm was working very well. Secondly, you see that outside these glandular shapes, most of the cells are green, which means they are not cancer cells, which is how it should be. And then within the gland, you will see that if it is blue marking, that means it is these blue dots are HER2 negative cells and the red dots are HER2 positive cells. Now compare that to the top panel. HER2 positive has this intense brown staining, which we have detected very well in the HND stain images. And if the brown staining is not very intense, that means it is HER2 negative, which also we have detected nicely in the images that you see in the bottom. Does that mean that it is 100% accurate? No, it is not 100% accurate. We have shown results where it worked very well. There are also cases in which it did not work very well, and no machine learning algorithm will be 100%. So it brings up a lot of ethical issues also, that even let's say if your machine learning algorithm is accurate for only 80% or 90% of the cases, then first of all, uh, is there some responsibility on someone to cross check the output of the machine learning models? Is there a responsibility to make machine learning models better, that even when they are wrong, they are able to at least say that I'm not confident about this particular patient, so please follow up on this patient with a more expensive test? So those are the kind of questions in research that people will be grappling with in the coming years. And it's also a question in ethics and law about how to introduce these machine learning algorithms into the clinic. See, it's a double-edged sword, right? If we don't introduce these algorithms, then overall as a society we'll have worse health outcomes. But if we introduce them, then we might become too reliant on them, and for at least a small subset of patients, we will make mistakes by relying too much on algorithms. So the question is, where is the right balance? So that is something that we'll be grappling with in the coming few years. Same with self-driving cars, for example. So if your self-driving cars become on an average better than a human driver, so for every million miles driven, a self-driving car will have fewer accidents than an average human. Okay, so, but then even when it makes mistakes, especially for those cases where it shouldn't have made a mistake, it seems very obvious. You might have seen in the news that a Tesla car ran into a truck because the truck was too wide and the algorithm got confused that it is not seeing anything. It couldn't see the truck because the algorithm was not, a human would have seen it, but the algorithm couldn't see it. So in that case, is it should we shut that program down, or should we go ahead with the comfort that at least on an average it is doing better than a human? So those are the kind of questions we'll be grappling with in the future. Any questions so far?
Sir, we have requested participants to write their question in the question answer session.
Okay. Okay. So I will
think there
finish
are
and then.
two. That will also be okay. Yes.
Okay. So I'll finish and then we'll.
Okay, sir. Okay.
Yeah. Okay. Okay. So here we are just showing that the genomic profile of the patient and what we detected using deep neural network, they match. So here, when we are looking at basal cancers, the probability of basalness is high from computer vision, and when we look at luminal breast cancer, the probability of luminal detection by the neural network is also high. So question is, so before this, what we did was we actually hand annotated the regions of cancer and hand annotated regions of HER2 positive and HER2 negative breast cancer, which is all nice. But what if we cannot annotate it? What if the mutation is too spread out to say that it is precisely at this location or that location? So can we teach a machine to not only detect the mutation, but also find out where exactly the hotspot of the mutation is without us telling it? So this is what we try to do with BRAF mutation in thyroid cancer. So here we use a special kind of neural network, which is called weakly supervised learning. In weakly supervised learning, we give it the entire image without telling where the mutated portion of the cancer is. Okay, we just give it the entire image. We just tell it that somewhere in this image, in this large image, there is a mutation, or nowhere in the image there is any mutation. So here, what the neural network architecture does is there is again like the layers that I pointed out, the convolutional layers and so forth. They detect a lot of features from all the sub portions of the cancer. And then there is another neural network on top of it that basically finds out whether I should pay attention to this region or not. So it makes a binary decision between 0 and 1. 0 means that this portion of the cancer looks like same in every patient. So it's not an important clue for me. Or this portion of the cancer looks different for BRAF positive versus BRAF negative patients. So here I should pay attention. So it basically, this neural network just computes the attention score for every location. And based on the attention score, we suppress that location or we enhance that location. And then we make a weighted average decision. And that weighted average decision turns out to be much better than if we just took an average decision over all the location. So the weight of the weighted average is computed by this attention neural network, which figures out whether the the location that it is looking at is has the same pattern everywhere, or does it have a unique pattern which is different between the positive and the negative patients. So with that, we got high accuracy. So there are some metrics that we look at in looking at accuracies. So let's say if you have, we don't just look at accuracy, we look at like something called AUC curve. So some of you might be familiar with it already. Uh, so suppose we are looking for tuberculosis in chest X-ray. It was recently in the news that there's a company in Bombay called Cure.ai that has done a very good job of detecting tuberculosis in chest X-ray. Now, let's say we look at a population of 1 lakh patients, and out of that, 90,000 patients or 99,000 patients, right? They have no tuberculosis. Only 1,000 patients have tuberculosis. So if I have an algorithm that has no intelligence at all, and it just says that no one has tuberculosis, then it will be 99% accurate just because only 1% patients have tuberculosis, which is not a desirable outcome, right? We want to be able to catch patients for treatment who have tuberculosis. So just looking at accuracy is not enough. We should look at something called both sensitivity and specificity. So sensitivity means if the patient is positive, then what percent of times we catch them, and specificity means if we catch them, what percentage of them are actually having the disease that we wanted to catch. So we look at the two metrics together, excuse me, and plot it into a curve. And the area under that curve is what tells us how good that algorithm is. So that came out to be very high for BRAF positive detection in thyroid cancer. Not only that, if you look at the tissue, that attention map that I was talking about, attention calculation for different locations, that gives us a heat map. And that heat map tells you whether it is finding regions that are positive of thyroid cancer location, sorry, BRAF mutation or not. So red means positive, blue means negative. So there is very little blue because this is mostly a BRAF positive patient. And if you look at a special test on the same patient, we will see that it is more brown. Again, this brown is the IHC that detects BRAF mutation. More importantly, when we zoom into this area, you will see on the right hand side, that's a zoomed in version of that. You'll see these black dots are the cells, and you will see a certain pattern of the cell shapes. And that is called oncocytic cells, and that has been previously detected by some pathologists, and they had thought that it is linked to BRAF mutation, but they could not prove it. They had a hunch, but they could not prove it. Whereas if you see on the BRAF negative tissue on the bottom panel, here you will see that the attention is mostly negative, which means that the tissue pattern here is different. It is more indicative of absence of BRAF mutation. And when we zoom into these orange portions and we enlarge it, we see that that oncocytic pattern that you see on the top, you see this pink bubble around the nuclei that is missing. So the oncocytic pattern is missing. So this confirmed what pathologists in 1950s and 60s had a they had an intuition that this may be associated with BRAF positivity, the oncocytic feature, but they could not prove it because it required statistically that they are able to reproduce their findings, and they couldn't reproduce their findings because it is human intuition is not replicable from one observer to another observer, right? So that's why we have some very uh, well respected experts whose knowledge cannot be easily passed on to another human. But that doesn't work for mass scale diagnosis of diseases. For mass scale diagnosis of diseases, we need replicable observations, and that is where we think that some of these works that we are doing and several other groups around, across the world are doing, that is going to help in making replicable algorithms to find these patterns and detect mutations. So what goes behind the scenes of this is that we have a lot of different algorithms that help us get to this stage. One of the algorithms is something called color normalization. If you look at the top two uh, images of cancers, you'll see that the left one is more pink and the right one is more red. So this happens because the chemicals that are used to give these colors, although they are supposed to be purple and pink, they are slightly different between different labs. So, for example, someone may order chemicals from a different vendor. The other lab may order chemicals from a different vendor, and the color of the chemicals is slightly different. The other thing that can happen is that the technicians in the lab are trained differently, so that's why someone puts it in the slide in the chemical for too long, someone doesn't put it for too long, and so forth. Because of that, the color intensity changes. And then when you take an image of that slide, right? So depending on what camera sensor you use on the scanner on your cell phone, the colors come out to be slightly different. Now, this change in color is actually not a problem for humans. We can see that, okay, there is a change in color, but I need to concentrate on the shape. And because of the shape, I can tell which one is grade 1 cancer and which one is grade 2 cancer. But that is not so for machine learning, because machine learning is learning directly from the image without us telling us what to look for in the image. It gets confused when the color pattern changes. So to increase its accuracy, what we do is something called color normalization. So if you look at the image on the bottom, it is the same as what is on the top left, but using the colors of the top right image. So we preserve the structure of the top left image, but give it the colors of the top right image. So that is called color normalization. So this is the work we did uh, with this Technical University of Munich in Germany. And this is an outline of the algorithm. So we first take both the images, and we separate their colors using something called stain separation, using something called non-negative matrix factorization. Then we take the structure of one image and we give it the color of the other image, and we recombine it. And then you see that the structure of this top image is preserved, but it is given the color of the bottom image. So the source image remains the source image, but it takes the color of the target image. So this algorithm that we published and the code that we released, it has been used by several people now across the world, and they are quite happy with the results. Some people have improved upon our algorithm have published new research with slightly better algorithms. So because of this, because of color normalization and so forth, our next steps such as nucleus detection works quite well. So if you look at the image on the left, all the black dots that you see are nuclei. Now, for some of the things that I talked about, for example, detecting cancer, then finding whether it is HER2 positive or HER2 negative cancer, we concentrate on these nuclei. Now, detecting these nuclei is not an easy job because you will see that some of these nuclei are light, some of them are dark, some of them are have a hole in the middle. Looks like what is a hole in the middle. So if I don't know if I can zoom in. No, I guess I cannot zoom in. But you will see that some of them have a light hole in between. So that creates a havoc for detecting these nuclei as separate entities. So, but now using deep neural networks, we are able to detect pretty much every nucleus that you can see we have marked with a green dot on the right image. Here's another view of the nucleus. This is much closer. So you can see that, for example, if you look at this nucleus, there's a white blood cell which has been dyed by hematoxylin dye. That's why it looks like it is very dark. And this one is very easy to detect because it's a dark blob. Whereas this cell, you see that its darkness is only on the boundary, and inside you will see there's a white space. So it's very difficult to find this as a separate cell. But with these deep neural networks, we are able to detect each and every cell, including cells that are touching each other, as two separate cells. So this again is we prepared a data set of all the cells and their boundaries marked. Some 20,000 cells we hand marked, and we released that data set for the world to try their algorithms on, and we also released our own algorithm. So that data set has also become quite popular. It is used by a lot of people to train their deep neural networks now for cell detection, for nucleus detection. And we organized a competition based on that in an international conference, and which saw a lot of participation from different people. And and it has become quite popular now. So then we did a follow up on that competition. We said, okay, not only detect the cell, but also classify the type of cell. So epithelial cell is the tissue, is the gland cell where the cancer starts. But once the cancer starts, what happens is that the immune system tries to stop it. And so immune system brings white blood cells, which are lymphocytes, macrophages, and neutrophils. It brings those to attack the cancer. Sometimes it attacks the cancer. Sometimes the cancer reprograms the immune system to help cancer develop faster. So that entire mystery of whether when you see immune response, whether it is trying to kill cancer or whether it is helping cancer, that mystery has not been fully resolved. But in immunotherapy, what they are doing is they are trying to reprogram the immune system to attack the cancer better and circumvent the defenses of the cancer against the immune system. So, in general, basically, suffice it to say that to study the cancer and the presence of different types of immune cells around it is important to unravel these mysteries and to find out the effectiveness of immune therapy. So we had marked some 46,000 nuclei and got it checked by a pathologist, where we showed where are the epithelial cells, where are the lymphocytes, where are the macrophages, and where are the neutrophils. And we again conducted a competition where research groups from China, US, UK, and several other countries they participated, and these are some of their results. So these are four different organs for which we mark the cells. The second row that you see with the blue and the green and the yellow and the red are the four different types of cells which we had hand marked. And below that, you will see what the algorithms from these TIA lab in University of Warwick, STJU in China, and this one in Iran, they detected. So what you can see is that their detection is very close to the ground truth, which means that these algorithms are becoming very, very good at detecting the boundaries of cells, even if the cells are touching each other, even if cells are very different in sizes from each other and very different in shapes and appearance. And even if they are very crowded or not. So with this, now people can start using these algorithms to actually study the immune response to the cancer. Another thing that we are doing is that we are trying to model all the cell nuclei. So this is the middle bottom that you see is that detection of all the nuclei that I had shown earlier using a neural network. So, but when you detect all the nuclei, you can see that each nucleus is influenced by its neighboring nuclei. So whatever is influencing that nucleus, if you join them with a line, you will form a graph. So each vertex of a graph where the lines meet is a nucleus, and the line is the sphere of influence of that cell, whether it is being influenced by a neighboring cell or not. So you will see a graph like this. So this study of graphs is uh, is important for many fields. So for example, if you look at the social media graph, right? So whoever are your friends or followers on Twitter or Facebook, and whoever are their friends and followers and so forth, that all forms a social media graph. So if you look at a graph of social media, you can find who is the main influencer in a group, right? So among you and your friends, maybe one person is more opinionated, one person does more thorough research about political issues or social issues and post something on social media, and you get influenced because their logic sounds good to you. And not just you, but several other friends get influenced, and then you forward that post to other people. So that community structure within the graph starts to form, and then you can see that and you can analyze that, and based on that you can. So let's say if I was an ad company and I wanted to influence, reach more people by spending less money, I will find the influencers and I will influence them first, instead of influencing a single person who's very asocial and who cannot influence other people. So the same kind of logic helps in cancers. So if you look at the graph of all the cells, then there are some cells that are more influenced or or they influence others more. So and then we can see that whether a cancer is surrounded by different types of cells, such as the different immune cells that I talked about earlier. So mining those properties of neighborhood structures in a cancer community helps us characterize whether this cancer is easily treatable or not treatable. So there is something called graph neural networks whose input is a graph, and then it has convolutional layers slightly different from the convolutional layers of the images, but similar concept. And based on that, it gives some output about, we train it to predict whether the cancer is treatable or not, or if, whether the cancer is of type 1 or type 2. So here we are looking at two different types of cancer: in situ breast cancer and invasive breast cancer. And you can see the steps that we took. We first uh, took the image, we detect all the nuclei, and then based on that, we mark which nuclei are important to classify it into in situ versus invasive cancer. The difference between in situ and invasive is that you will see these sharply defined boundaries in situ, and it's a less dangerous cancer because the cancer hasn't broken out of this boundary. Whereas when it has broken out and it has spread, then it will look like a pattern here, which is the invasive breast cancer. So you can see that the attention of the neural network is high, which is these orange dots at the boundary itself and not at the inside. So which means it is trying to now find the features that are important in the cancer to be classified as in situ breast cancer. Similarly, in prostate cancer, there is something called Gleason 3 and Gleason 4. Gleason 4 being more dangerous. And there's a similar concept that if you see these circular gland shapes, that means it is less dangerous. That means it has not become too chaotic yet. So here again, with that attention mechanism on the graph, we see that it is able to detect all these boundaries of the glands, whereas it doesn't show much of that in the Gleason 4 prostate cancer. Then this is another color normalization algorithm where we use neural networks to do the color normalization so that images from two different uh, labs look similar. And then we also start to work on things like active learning. So annotating and labeling images is very expensive. So for example, let's say I want to I have a repository of 1 lakh slides in Tata Memorial Hospital, and I want to detect their mutations. Now I cannot do a mutation test for 1 lakh cancers because that is very expensive. What if I can only, I have budget for only 1,000 of them. So which 1,000 should I pick for labeling? So instead of randomly picking 1,000 out of 1 lakh, what if an algorithm intelligently picked those that look new to it? and said that, okay, take these 100, label it for me, and then I will find out which ones are still new for me, and I will give you another list of 100 to label, to do a genomic test. So that is called active learning. So here we developed an algorithm that did very well compared to what the previously published algorithms, where the accuracy for the percent of labeled data, our algorithm is the red line, is slightly above all the previously published algorithms, especially on this data set. So now we want to extend it to labeling of cancer images. Another type of algorithm that we are working on is called robust classification. What that means is that not only should an algorithm learn to distinguish between class A and class B of images, it should also say that among the class A, these ones are difficult and most likely mislabeled. So why that is important is, if you take a cancer slide and carve out many sub images from it, there are going to be sub images that are not useful, because because not every portion in the slide has cancer. Some could be fat, some could be stroma, etc., etc. So those are difficult for it to classify, or useless for it to classify. So it should be able to detect that. So by modeling the distribution of these patches, we are working on some other techniques where you see the images on the left are the ones that are not useful, and the images that are on the right are more useful. And you can see that the ones on the right are more densely packed with cells, and all of these are cancer cells. So whenever it doesn't see a cancer region, it finds that sub image to be not useful. And based on that, when we average the results, we get better accuracy than if we just average across all the patches, all the sub images. Other things that we are working on is looking at the cancers in multi-resolution manner. So if you look at the top right image, it's a image of a lymph node, again HND stain, and you see this slightly light region here, slightly light region here. These are germinal centers, and presence of these germinal centers in breast cancer lymph nodes that are close to breast cancer are indicative of whether the cancer is spreading or not. So these are indicative of what is the immune response to the breast cancer. So our job is to detect them well and to be able to segment them well, so that we can count them, we can find their area and so forth. So now this is a multi-resolution problem, which means that you don't only look at this image that you see on the top right, but you also zoom in and out to confirm whether it is a germinal center or not. Not everything is apparent from one particular magnification. So for that, we gave that zoom in and out capability to our neural network itself. It it has several blocks that downsample the features, and then it has several blocks that upsample the features. Uh, so this has been proposed previously as something called a U-net because of its U-type shape. But within the block itself, we have multiple resolutions themselves. Because of that, we are able to do a good job of finding all the germinal centers. So this is what it should have been like, the middle image, and the bottom image is what our detection is with the how it is different from the ground truth. And you can see there's a lot of overlap between the two images. So that means it is doing a good job, practically usable. The other thing is, how do we teach neural network something about the images without giving the image any label? So to take, for example, chest X-rays, let's say you have a repository of 1 lakh chest images, but you don't know whether they have tuberculosis or not. Only for 1,000 of them, you know whether there is tuberculosis or no tuberculosis. So, but you still have 1 lakh images. By a human looking at them, will start to see patterns that ribs look a certain way, heart is in a certain way, and usually most of them are clear in the lungs. Only some of them have some opacity in the lungs and so forth. So can the neural network also learn something from images without being told what it is looking at? So the way we do it is we do something called self-supervised learning. We take these images and we create a jigsaw puzzle out of it, and we jumble the jigsaw and teach the neural network to unjumble the jigsaw. To unjumble the jigsaw, it learns the structure of the ribs and the spine and so forth, so that it can put them in the right order. And in doing so, it teaches itself something about how these images should look like. Now it becomes easier to train it to find tuberculosis versus non-tuberculosis with the fewer samples. So here, this is an example of right ventricle segmentation in MRI and in heart. And what you can see is that if you do not teach it the jigsaw pattern, then it requires more number of samples, which is on the x-axis, to catch up in the accuracy. Whereas if you start with teaching it first the jigsaw and then you train it to segment the right ventricle, then it starts with a higher accuracy. Eventually, both of them catch up. But if you have fewer samples, then the blue curve is better. That means solving the jigsaw is better than starting with some other neural network. So that's pretty much the end of my presentation. Are there any questions?
Hello.
Yes.
Yes, sir. Actually, if you look into the Q&A section,
Yeah.
or uh, should I read it for you?
Where should I start? I see a lot of them.
No, this is a chat box. First,
Yeah.
you just check the Q&A section,
Q&A
just
section.
besides the chat box symbol. Yeah,
Q&A. Okay, I found
correct.
it. How to define the size of the cancer images? So Digambar is asking how to define the size of the cancer images. So these slides that you get from pathology, these are very large, right? So they are as large as I mentioned, 50,000 by 100,000 pixels. So definitely you cannot work with the entire image in one go. So usually you have to cut them into smaller numbers. Easiest size to define is whatever is a standard neural network size. So there are some standard architectures such as ResNet, and it takes images of size 224 by 224. So chop this up into sub images of size 224 by 224. That's the easiest answer. But as you gain more experience with working with these neural networks, you will start to use your own architectures, and then you will realize that perhaps to see the context of the cells around it, you might need a bigger size, such as 1000 by 1000. So it all depends on the type of problem that you're solving. Sir, how were the heat maps generated? Aisha is asking. And which tool was used to visualize the clusters in the 5D space, 50D space? So the heat maps were generated. You take these sub images on different portions of the slide, and for each sub image, you generate one color of one location of the heat map, and then you move to the next location and process it again through the neural network. So then for each patch, you have a different color, and you put all those together, and it becomes a heat map. The tool used for the 50-dimensional visualization is called T-SNE, T-SNE. So just search for T-SNE, you will find that tool. Prem is asking, I understand you are talking of optical images. What kind of magnification is used in generating these images? So the magnification is around 400x. So you have a 40 piece objective and a 40x objective and a 10x eyepiece in the microscope. So it's equivalent to that. But to be more precise, a quarter micron by quarter micron of a tissue sample is mapped to 1 pixel of the image.
Okay, okay, that's next.
Thank you,
That's
Mr.
nice.
Premgoya.
And my second part of the question.
My main question is whether the present image analysis methods can be used to test the efficacy of cosmetic creams. That will help public to decide whether to believe in the advertisements or no. Yes, so I believe it can be tested, but it will require careful definition of the problem and data collection. And then after that, we can do a scientific test. We can have a case and a control group. Control group doesn't use the creams, and case group uses the cream, and we can see after 1 month whether there's an impact or not.
So is there some work already done on this line? Is there some work, you are aware of any work being done
No,
on
sir,
this?
I'm not aware of any work, but I think there will be some effect because a lot of these creams have bleaching agents, so they actually have some temporary effect.
Right.
But after that, after you stop using the cream, the skin reverts to its
So is it
original color.
So they talk of softness of the skin and the things of that sort.
Right,
So
right. So that also can be assessed, but I'm not aware of any work that has been done.
thank you, sir. Thank you.
How to decide which deep learning technique is suitable for the data? So a lot of that you can get that intuition by reading papers and seeing whether the inputs that were used in that research paper, are similar to the inputs that you are using in your research. So based on that, you can develop some intuition. And a lot of it is, there is no easy answer. You only have to try and fail at all the methods that didn't work until you get to the method that works. Sir, may I know the design flow that is adopted? So to answer Vivek, the design flow that you adopt is, first you define the problem that I want to classify between class A and class B and make it very, very narrow. That within invasive ductal carcinoma, I want to classify between HER2 positive and HER2 negative invasive ductal carcinoma. And I want to do it only for 40x magnification, not for any variable magnification. So you make it narrow, and then you collect the data. So they collect the data such that you have at least 20,000 small images for each subtype. And then you train a neural network to classify between them. So you need a lot of data for that. And then you also take some patients on which you did not train for testing. Only then you will know whether your algorithm worked or not. Otherwise, these neural networks are so big that it is very easy to overfit on the data that you have. So NN for CUDA, yeah. So neural network for CUDA, CUDA, not KUDA, CUDA. So best way to start would be search for KERAS, KERAS.
KERAS. Yes, sir.
Yes.
Okay.
So that's the easiest library to learn.
Okay. Okay. Thank you, sir.
It comes with TensorFlow. Kindly enlighten research topics in the field of machine learning. So the research topics in machine learning it's a very vast subject. So for example, better active learning techniques, better self-supervision techniques, better weakly supervised learning techniques, machine learning models that have a good idea of their own confidence. That is another research topic. Then finding new applications of existing machine learning methods is also a good research topic, and that's probably the easiest one to get to. So for example, let's say you work with some doctor and they have a lot of skin lesion images, right? So whether people have acne or they have something else and so forth, and you can train something to classify between, let's say, different kinds of skin lesions, different type of oral lesions for leukoplakia, erythroplakia, because oral cancer is a big problem in our country. Uh, you can work on detecting defects in parts, for example. So you have a nut and bolt factory. You have nuts and bolts that have no defects versus those that have defects. You can train neural networks to classify between those. So in general, application is easier to work with.
Yes, sir.
Uh,
Sir, IoT base,
yes.
can we do machine
I'm
learning
sorry.
with IoT base?
Yes, you can do it with IoT also. You have to find good applications in IoT. You can work there also.
Yeah, thank you.
Then Miss Vijaya is asking, sir, age-wise cell density changes. How to claim accuracy and sensitivity of work, even with deep learning algorithm. Some cancers stop growth in between, so how to justify work? So yeah, usually to define these problems, a machine learning person cannot do on his or her own. You have to work closely with a medical doctor because they will interpret the data for you. They will help you formulate the problem better. Without them formulating the problem, without them helping you define the problem better, it will become very difficult to do this. So all these kind of questions that you are asking, these will come up if we don't define the problem narrowly. So currently machine learning only works with narrow problems. So let's keep that in mind. Deep learning neural network is a black box. How to justify the decisions? So that is a very good point. Deep neural networks definitely are a black box. We don't understand how they make decisions at a granular level. So to justify their decisions, you just have to test them very rigorously. And if they work rigorously in varied conditions, only then their use is justified. That's the only way to justify it. So, and then another example of that would be that we trust certain things in very complex situations. So, for example, autopilot of a airplane. The airplane autopilot has not seen all the routes and all the weather conditions. Yet it has been tested enough that we trust it. We justify the decisions of the autopilot. So the same kind of thing has to be done in machine learning also. But in autopilot, there is always a pilot present who can take over when the machine is making mistakes. The same kind of logic will be required right now for machine learning as well. Meenakshi ji is asking why you use genome dataset. There are many datasets openly available. So yeah, genome dataset, we wanted a large dataset, and that TCGA dataset is quite large. So first we wanted to define heterogeneity at the genomic level. Then we wanted to recognize it using images. So images is where I mostly work. I just did it for the genomic test because no one else had done it, and we didn't know how to train the image detection, image classification, unless we define the problem at the genomic level. So that's why we had to go to the genomic level. What is the difference between normal CNN and graph CNN? So normal CNN, the input is an image. In graph CNN, the input is a graph. That is the main difference. Otherwise, inside the structures are slightly different, but that is a less important difference. The main difference is that type of input is different. I don't give the cancer image to the graph CNN. I give, after I form the graph, after detecting all the nuclei and finding the neighborhood structure, I give the graph as an input to the graph CNN.
Sir, there are few questions in the chat box. Should I read it for you?
Yes, please.
Which software can be used to study nanoparticles and ML?
So nanoparticles, once you have images of nanoparticles, again, you can use Python, TensorFlow, and Keras combination. But after images, it doesn't matter. After you have gotten the images, it doesn't matter because they're all the same images, whether they're cancer images, images of faces, or images of nanoparticles. After that, you can use the same software to study all images, which is Python, TensorFlow, and Keras.
Okay, sir. What parameter to identify in cancer detection? Generally, it will identify in the third stage. Any detection before the third stage?
Yes, so there is a research going on to actually predict before the cancer strikes. So the cancer does not happen in a day, it happens over years. So some abnormality in the tissue will start before it actually becomes cancer. So let's say if we were taking mammograms or lung CT for a large population and we did it for 5 years in a row. So let's say we did it for 1 lakh patients and we did it for 5 years in a row. Out of 1 lakh, 100 of them become cancerous within 5 years. So now for those 100, we know that what the pre-cancer looked like years before it became cancer. Now we have a hope of training machine learning to spot that in other people going forward before it becomes cancer. So if we have cancer prevention drugs that come into the market, they will be a test available to detect pre-cancer before it becomes cancer.
Okay. Sir, which mining algorithm is best to extract frequent patterns from big dataset?
So it all depends on the type of the data. So if you are using image data, then CNNs are the best. And for CNNs, the software combination that I talked about is Python, TensorFlow, and TensorFlow now comes with Keras. So basically Python and TensorFlow. And there are lots of tutorials available online to get started with this.
Okay.
There's a question, what is the meaning of a graph? So graph means that your input is not like an image. It's like a set of vertices, right? So vertex is basically, let's say the location of each cell, or it is one person in a social network is a vertex. And edges means pairwise combinations, which is which two people are related or which two cells are close to each other. So pairwise relations. So that is a graph.
How to identify patches?
So basically, you just run a program that goes, let's say you have a very large image, it takes this sub image, then it takes the next sub image, non-overlapping sub images, and those sub images are patches. And then you can write slightly more intelligent algorithm to get these patches, where you see whether the patch is all white or all black, then you reject the patch, you don't store it. Because if it is all white and all black, that means it doesn't have any cells, and it doesn't require any analysis. So you can intelligently find only those ones that are neither completely black or nor completely white, and then keep only those as inputs for your neural network.
Okay.
Do you use malignant tissues for your work? Yes, these are all, a lot of these were malignant tissues. Is it possible to go to root cause with the help of graph? Yes, that is our hope, that we will be able to go to the root cause, but it is not always possible. Some of these problems are quite difficult for machine learning to solve also. And but with large data, we hope that we'll be able to do that.
Any other question from participants? We'll wait for a few
Sir,
minutes, sir.
Yes.
which type of input images is are of which stage actually? Stage 1, stage 2, and whether it
Yes.
affects on the accuracy?
Yes. So it does affect. So we try to control for the stage also. So we train separate algorithms for, let's say, stage 1 and separate for stage 3, because their treatment will also be different. So we work with both stage 1, stage 2, stage 3. We work with all of them, but we try to control for that also, because we don't want the stage to be the main determining factor.
Okay,
We
thank you,
want to
sir.
get, yeah, more intelligence, more insights than just stage, in addition to stage. So these tissues are actually, they are not in my possession. These are with their respective hospitals. They keep it for a long time. They keep it for 10 years, for example. So I just get the digital images of those tissues. And once the digital image is put in a data center, then it can stay there for perpetuity, for as long as we want to keep them.
Any other question?
Amit sir,
Yes,
as you just mentioned that mutation is a continuously going on process.
right,
Okay, so in some cases that mutation may result into the cancerous growth.
right.
So is it mutation
Hello,
is to be just
I couldn't hear you for the last few seconds.
what is the basic reason that some mutations may just turns into that cancerous growth?
So whenever a cell becomes mutated, usually that mutation is unstable, and because of that, the cell naturally dies. And if it doesn't naturally die, then the immune system kills it off. But if the mutations become too many, then either the immune system gets overwhelmed, or what happens is that some mutations are so uh, different that they actually bypass the immune system, the attack of the immune system, or they even reprogram the immune system to help itself. So it's just like how mutations have given us the evolutionary advantage to become human from a primate. Similarly, the same type at a micro level is happening with the cells, that is giving them advantage over the rest of the cells to become cancer. So part of the reason we have more cancer now is because we are living longer. We are not being killed by tigers. We are not being killed by diseases. So as we live longer, we accumulate more mutations and our body becomes unstable, or some of the cells become unstable. The other problem is that we have more influences on our body that we were evolved to see. For example, we were not evolved to see chemicals, electromagnetic radiations, right? Pollutants. Those we were not evolved to tolerate on our body for the millions of years that we evolved. So suddenly, because of our industrial environment, everything has changed, and our body hasn't evolved to keep up with that. So it is accumulating more mutations.
Sayed sir, you can unmute yourself and ask. Sayed sir, you have raised your hand. If you have any question, please unmute yourself and ask the question.
Yes. So Mr. Bhattacharya is right. Mutation is not only causes cancer, it also changes heredity changes, and it also causes bifurcation in species. For example, so there was a common species from which all the primates, including humans, evolved. So that bifurcation also happened because of mutations. Hereditary differences are also because of mutations. So not all mutations are harmful. Only some of them are stable and harmful to become cancer.
Sir, I request you to share your presentation. You can direct to.
Hello, sir. Good afternoon.
Hello, good afternoon. Yes.
Sir. I wanted to ask you, how do you label images?
So that is done in collaboration with a medical doctor. In
Okay,
collaboration with, yeah, we talk, I have a collaborator friend in Tata Memorial who's a pathologist, and then in University of Illinois Chicago he's an epidemiologist. So and then another one who's in King's College London, she's a molecular biologist. So we work together.
yes, sir. We also working on microscopic image analysis, so that's why I asked you.
Right. Yeah, as an engineer, I don't have a lot of this knowledge that these people have, and as medical doctors, they don't have the knowledge of making AI systems. So we work together.
Okay, sir. Thank you, sir. If we require any help, could you please help us in future?
Yes, you can email me at amitsethi@gmail.com.
Okay, sir. Thank you, sir. Have a nice day.
Thank you.
Sir, sorry, sir. I meant that please share your PPT file with me so that I can share
Oh,
it.
okay.
Yeah.
Okay. I will share it with you.
Yes.
Okay.
I think we can wind up here.
Okay.
If there are any questions, I think people can reach you by email.
Right.
Can you share, type your email ID in the chat box?
Yeah.
Okay. Thank you, sir.
Okay. Thank you.
Let me present my screen.
Okay. I will drop off then. Okay. Thank you, Mr. Deshpande.
Sir, 2 minutes, please.
Okay.
Okay, is my screen visible?
Yes.
Sir, thank you very much for accepting our invitation, first of all, and having here with this informative session on biomedical applications in machine learning. Please accept a virtual memento as a token of gratitude from us.
Okay.
Thank you, sir. Thank you very much.
Thank you. Okay.
Thank you.
It was a pleasure.
Thank you.
Thank you, sir.
I request all the participants who are having any issue related to login If anything is there to discuss related to login issues, please be here so that we can sort your problems. Regarding attendance, we'll be taking your attendance. You don't have to put your attendance or any Google